Am I live? I am live. Alright. So, I'm just gonna wait a bit longer, let some people come in, and then I'll get right into it. Alright, I'll just get started right now. So hi, my name is AJ. This is going to be a presentation about the biology portion of the MCAT. It will be covering every single chapter, every single concept that you're going to need to know to do well on the biology section of it. It won't be covering like the super detailed nitty-gritty stuff that you might see in like a Princeton review book or a Kaplan book or whatever, but it will be covering like the big like picture concepts, everything that you are guaranteed to see on test day. So with that being said, I'm also going to be doing similar presentations like this with biochemistry, physics, chemistries. I won't be doing one for cars as there's not much I can really teach you for that. You just have to read faster and comprehend faster as well. For psychology, I won't be doing one either. I think the Khan Academy doc, which is like about 300 pages long, is more than enough for the psychology section. So I think just studying that as well. That being said, this series of lectures will be dedicated towards the April 9th test takers. So I'm going to do my best to make sure I get these out to you guys as quickly as possible. All right, with that being said, let's just jump right into it. We'll be focusing on the first chapter of biology, which is the cell. The cell is probably the most important chapter that you should be covering. It's 1/5 of the questions you'll be seeing on test day, so I highly recommend just like learning this chapter, getting into the dirt with it, knowing everything like front and back, learning like the back of your hand. Let's start off with the organelles that are present in the cell. I think most of you guys will know like the organelles. They're pretty straightforward: nucleus, nucleolus. You should know what the structures are and their functions as well. Rough endoplasmic reticulum, ribosomes. I think all of these are pretty straightforward except for maybe peroxisomes. I feel like that's something like a lot of people just overlook, so I'll cover that really quickly. Peroxisomes are known for primarily periosomes. Sorry about that, guys. Peroxisomes. So they're known mainly for being like organelles that hold H2O2, hydrogen peroxide, and they play a major role in oxidizing beta acids. So I think just knowing beta oxidation is something that will take you guys a long way with peroxisomes. And they also play a role in the pentose phosphate pathway. I'll get into that in the biochemistry lection, but for our biochemistry section, well, for now, just know that they do play a role in the pentose phosphate pathway. And as for that, I'm not entirely sure what else I can talk to you guys about with organelles. Do you guys have any questions? Yeah, sorry, I'm just looking at chat right now. All right, no questions. So I'll just move forward with the organelles. One thing I'd like to talk about is like what is the pathway of a protein? Like where are proteins synthesized and where do they go? All right, so I'll get right into it. So proteins obviously start in the nucleus. It goes through transcription, translation, and then it will go out to the cell. So transcription takes place in the nucleus, so we'd start right here. And then it will be transported outwards towards the ribosome. So it'll be going towards the rough endoplasmic reticulum, as well as the ribosomes here, to be taking part in translation. Once it enters the rough endoplasmic reticulum, it'll be then transported to the smooth endoplasmic reticulum, where it'll be packaged into vesicles. Oh, sorry, I'm getting ahead of myself. It'll be transported after that to the Golgi apparatus, where then it is packaged into vesicles, and then it is released outwards into the cell. Now, where it can go from here, they usually go towards the plasma membrane. However, some of them also can go back towards the mitochondria. Some of them can go towards the centrosomes. Some of them go towards lysosomes as well. So I think the three ones that you need to memorize for sure would be the plasma membrane, the lysosomes, and the mitochondria. So moving on, let's go towards this picture right here. Can anyone tell me what this is? Yes, this is the fluid mosaic model. This is a very important model. You guys need to know this Cola too as well. Now, for this section, I won't be going into the details of like all this crazy stuff you're seeing right here, like the carbs, the alpha helix proteins, glycoproteins, globular proteins, the channels. You need to know this, don't get me wrong, but I won't be going over that today. Instead, I'll be going over these right here. Now, what am I drawing? What part of the fluid mosaic model am I drawing? This is the phospholipid bilayer right here. Now this part right here, hang on, I'll switch to a different color. This part right here, this is the phosphate head. This is hydrophilic. And these are the fatty acid tails of the phospholipids, and these are hydrophobic. Now, why am I talking to you guys about this? Why is this so important? God, it's so weird. All right, so why am I talking about this? Why is this so important? So the hydrophilic portion of the fluid mosaic model is very important because that is what allows the outer portion and the inner portions of the cells to interact with the membrane itself. So if you have hydrophilic molecules present on the outer portion, you increase the hydrogen bonding that can occur on the surface. More hydrogen bonding means more interactions with the outside world, which allows more proteins, more ions, more everything just to go in and go out. That's why it's present on both sides. So you can interact with everything that's going both inside the cell and going outside of the cell. The phobic portions are mostly there for structure. They obviously won't be showing up here. They have to stay in the center or else no hydrogen bonding will occur. So with the phobic, just know that it causes a decrease in hydrogen bonding. That's why it's located in the center. I think that's all I want to cover with the fluid mosaic model. The rest I can mostly cover in the biochemistry lecture. I'm looking at my notes right now. Cell theory, I think everyone understands what cell theory is. You guys should be fine with cell theory. You won't even be tested directly on it, so it's not that big of a deal. Mostly common sense. But yeah, so excuse me. Let's move on towards prokaryotes. So prokaryotes and eukaryotes, they have a lot of differences. I think the biggest thing that we should talk about would be the difference in the DNA. So with eukaryotes, they have their DNA present in chromosomes. So this is a very poorly drawn chromosome on my part. Yeah, you know what? I'm going to read you out loud. That's embarrassing. I'm a student, not an artist. They look like this. You guys know what chromosomes look like. Like that. You know what? I'm going to redo that one more time. And let's say like this being connected here with the kinetic core, spindle fibers, and all that. So a chromosome, it's different with bacteria and prokaryotes because they don't have a chromosome or necessarily chromosomes. It's more like a circular structure, like this. So this is eukaryotes. prokaryotes. Another thing I'd like to talk to you guys about with prokaryotes would be their shapes. I think you guys would know which one is which. So this one, causi. This one, spirili. Bacilli. One more thing I'd like to go over with prokaryotes before I move on to the next section would be the difference between gram-positive and gram-negative bacteria. So gram-positive bacteria, they have a large quantity of this protein called peptidoglycan. Now, this protein right here is present in their cell walls. So when you go through staining with gram-positive bacteria, they will have very, very thick purple like blots present, and those are the peptidoglycan that is being stained. I don't think you guys need to know the specifics of gram staining in detail. Actually, no, screw it. I'll cover it anyways. So with gram staining, you initially add in crystal violet, and then you add in iodide. Or I'll write it down too. So let me make some space. I'll cover it like right here. So step 1 of gram staining is that you stain it with crystal violet. And then you add in iodide. Afterwards, you wash everything out with ethanol. And then you stain with safranin. Now, safranin is a pink stain, whereas the violet is obviously a purple stain. So, it's opposites. I'm so sorry. Yeah, it's opposite. So, iodide and then crystal violet, because crystal violet must attach onto the iodide. And ethanol is used to wash everything out, and then safranin will stain everything pink. But in grams positive molecules or grams positive bacteria, there will be large amounts of peptidoglycan. Those won't be washed out with ethanol or safranin, so there will be no pink staining. Instead, it will show up purple. Gram negative, it's exactly what you think it is. There's not as much peptidoglycan. There's almost none. So they will get washed out completely. Safranin will be present. So it'll show up as pink. Yeah, so that is the gist of the cell. Let's move on. Actually, let me ask you guys if you have any more questions. No? All right, let's move forward to the reproductive system. Oh boy, this is a rough one. This one is very, very memorization heavy. So I think let's start off with cell division specifically. Before I cover mitosis and meiosis, let's talk about interphase. What's going on in interphase, guys? Something about G1, G2? Yeah. So G1 says at the S phase to G2 to mitosis. That's basically the gist of it. No, make sure you guys remember that there is a G0 phase. It's similar to G1, but there's no synthesis occurring afterwards. It's just living its own life. So yeah, G1, that's you're increasing the organelle count, the cytoplasm content. You're just making the cell bigger for synthesis to occur. Synthesis is when DNA replication occurs. That's when you basically just make a copy of the chromosomes. G2, it's same as G1. You're increasing it even more, getting it ready for mitosis. And mitosis, we'll jump into it right now. So I don't like this diagram because prometaphase will not be like a big thing on the MCAT. No, it exists, but it's not something that you have to like know hardcore. So PMAT, let's talk about what that is. So prophase, that's when the nuclear envelope decondenses and the spindle fibers start forming. So you can see right here that the envelope is starting to going to be disappearing. Or that's what like pro and metaphase are part of. So that's disappearing. It's ever getting ready for metaphase, where everything aligns up on the metaphase plate. So everything's being lined up in the center to be split in anaphase. The spindle fibers will then tear the homologous chromosomes separately, and they'll be on their own sides, where they will be split into their daughter cells through telophase and cytokinesis. Cytokinesis is the splitting of the cytoplasm. Telophase is when they just pinch it off via cleavage furrow. Note that mitosis forms diploid cells. This would be 2N right here. Meiosis is similar but also different. Meiosis, it occurs in the same way. Prophase 1, metaphase 1, anaphase 1, telophase 1. But it's one important thing to notice that crossing over and disjunction occurs, specifically at metaphase 1. Let me mark that right here. Now, after telophase 1, it splits through cytokinesis, forming 2 daughter cells. These will then go through meiosis 2, which is the exact same as meiosis 1. Note that these cells form haploid daughter cells. These are not 2N, but 1N. So this occurs right here, forming, going from 2N to N, and then these two will just form N as well. That's how you get 4 haploid cells. Now, moving forward, let's talk about the reproductive anatomy of the human body. So let's start off with the male reproductive system. I think this is incredibly easy. You gotta know, what is it? The pathway of sperm, which is 7-up. Or let me write this a bit like further to the left so I can actually like write the parts. PMAT, 7-up. So before I say, does anyone want to tell me what 7-up it means? Like, what's the sperm pathway? Nobody. Okay. This is incredibly easy, guys. Let's go. I'll walk you guys through it. So we start off in the testes, which has the seminiferous tubules. So that's the first S. Seminiferous. I don't have space. All right, I made it work. Don't worry. So yeah, we start off in the seminiferous tubules. We travel up through the epididymis, which is the second part, epididymis. And we travel all the way up towards the vas deferens. That's the tube that's connecting us all the way up and around. So that vas deferens. The vas deferens leads us towards the ejaculatory duct. There's no fan. Don't worry about that, guys. It's just part of the acronym. Through the ejaculatory duct, it's reaching the urethra, and eventually it leaves as ejaculate through the penis. So yeah, that's pretty much all you need to know for the male reproductive system. Like, obviously, you might have to, if you guys want to brush up on stuff like the bulbourethral gland, the prostate, Like, all that's good stuff to know, but for the most part, it's just knowing 7-Up. So now let's move on towards the female reproductive system. This is much more complicated, guys. So this is like the base anatomy of it. If there's any like thing that's like, if this is the most bare bones, like you'd have to know. So the pathway of the egg upon fertilization is that it's in the ovary, it's fertilized in the fallopian tubes, it travels this way towards the uterine lining where it is then implanted, and then it will grow, and then eventually it'll form a fetus, and then eventually a child. So one thing that I'd like to talk about specifically are ectopic pregnancies. That's something that the MCAT really likes to test when it comes towards the female reproductive system. So what is an ectopic pregnancy, guys? All right, so nobody knows what it is. That's not a problem. I'll talk about it. So an ectopic pregnancy occurs when eggs normally are implanted in the uterus, right? Well, an ectopic pregnancy occurs when it's not implanted here, but it's implanted maybe somewhere in the fallopian tube. It's just anywhere that isn't in the uterus. And that's very dangerous because growth of the egg inside, or that isn't in the uterus, will end up leading towards growth. It'll lead to rupturing. It'll lead towards lots of internal bleeding. Very dangerous for the mother if her tubes get ruptured. Organs could be lost upon treatment. It's very risky. So you guys should know that an ectopic pregnancy is not a good thing. It's fatal. And that it has to be in the uterine lining in order for it to grow into a healthy fetus. Now, speaking of healthy fetuses, let's talk about some embryogenesis. So as you can see here, this is the pathway it takes, going from a fertilized egg cell to when it gets split, it becomes 2 cells, moving out to 4, 8, 16. After that, it becomes much, much larger. There's so many cells. It's called a morula. A morula eventually becomes a blastula, and then a blastula becomes a gastrula. Tongue twister. So blastula to gastrula. Morula, blastula, gastrula. Now, there are 3 layers to the gastrula. It's the ectoderm, the mesoderm, and the endoderm. I'll get into that in just 1 second, but make sure you also know a little bit about the anatomy of the 3-layer gastrula, because it'll be going in through a blastopore, which is sort of like a little hole right here, and there's also an arc aneron, which is like a little arc right there. So if they ever see these, just know that that's what they're talking about. So now the endoderm, ectoderm, mesoderm, they all develop into like the human body. They're like the beginning, they're the baby stages of the body. So the endoderm is the inner layer of the blastopore, or the gastrula. It's the internal layer, endo, and it develops into lung cells, thyroid cells, and digestive cells. The mesoderm is the middle layer, and I like to call it the mesoderm. Because it provides the means for you to travel, to walk, to move, to function. And that's where it comes it comes from the skeletal muscle cells, the cardiac muscle cells, smooth muscle cells, red blood cells. It provides basically everything you need in order for you to be able to move. That's why I refer to it as a mesoderm. I don't have anything cool for the endoderm, I'm sorry. Some people say it's like the endernal layer. That's not something that I'm a huge fan of. If it works for you, sure. So endernal. Sure. I'll rewrite mesoderm too. That's a bit rough. And then the ectoderm. So this is the attractoderm. This provides you with all the stuff that makes you attractive, like your skin, the skin cells, your neurons in the brain. Brains are attractive. Being smart is sexy, guys. Pigment cells, that also goes in towards your skin tone. It also goes into your hair cells. I want to point that out as well. So attractive derm right here. I think that's everything I want to cover with the reproductive system. There is I want to cover menstruation as well, but I'll be going into that with what is it, with not homeostasis, sorry, the endocrine system, along with all the other hormones. So with that, let's move on towards the next topic, which is homeostasis. Now homeostasis, this is a rough one, I won't lie. Where should I even start? Let's start off here, I guess, with osmoregulation. This is a picture of a nephron. This is the functional unit of a kidney. Wait, sorry. I'll just have to say, like, if you're not comfortable with the structure of a kidney or exactly what it does, you're gonna have to read the chapter over again. There's a lot of like little like inner like specific details, and I can't cover that in this lecture because it'll just go on forever and ever. But I'll just, like I said, just go over it in your chapters in your books. I'll just go over through the important stuff right here. So I want to point out specifically the glomerulus right here. This is where filtration takes place. So whenever like the fluids in your body enter the kidney, the nephron, the glomerulus and its surrounding Bowman capsule will take in all the fluid, it'll filter everything out, and it will mostly let out the various different ions, so it's like sodium, chlorine, potassium, stuff like that. That's where secretion occurs. Note that secretion is passive and active. Also, like to note is that reabsorption is the exact same. Or not the exact same, but it's similar in that it is also passive and active. So secretion occurs in the proximal convoluted tubule, where all the sodium, the chlorine will be released, water would also come out. It'll pass in through the loop of Henle, where more water will be released out, and it keeps on going up. And then it reaches the distal convoluted tubule, and it starts going right back down into the collecting duct and then through the ureter. So one thing I want to know is, I'm looking at a diagram myself. Yeah, I covered basically everything. So secretion occurs here in the proximal convoluted tubule. So I'll just write here, proximal. And reabsorption occurs in the distal convoluted tubule. So your sodium, that's gonna be messy. Yeah, I'm sorry guys, it's a bit messy today. Alright, you know what? We're just gonna say proximal, passive, and active secretion of sodium, chlorine, water. And then reabsorption will be the same. It reabsorbs sodium, chlorine, water. Where else do I want to go? Let's talk about, I'll get to the liver. I'll talk about the liver last, and I start with the kidney first. Let's go towards ADH. Now, where does ADH come from? We'll talk about it in detail more later, but let's talk about, let's just link it towards other parts of the body first. Where does ADH come from, guys? Right. It comes from the, well, I guess you're sort of right. It comes from the hypothalamus. What gland exactly am I talking about? posterior pituitary gland. So, that being said, ADH is also known as vasopressin. And its function is to increase the permeability of the collecting duct. Now, by increasing the permeability of the collecting duct, it allows more water to come in and then be reabsorbed to water. I'll write that. It allows most of it to be reabsorbed and it can be excreted out as urine. That's the main function of ADH. And aldosterone, it's the hormone that basically stimulates secretion. So this stimulates the sodium reabsorption, potassium, and hydronium. It's from the water molecules secretion. It also increases blood pressure and blood volume. That's, I think, all I want to cover with aldosterone. Note that it comes from the adrenal cortex and it's regulated by the renin-angiotensin aldosterone system. But that's not like something that the MCAT will ever talk to you about. It's just something that you need to know that it is regulated by various systems. And let's cover the liver last. So it has 7 major roles. So let's just jot that down real quick. So the first one is that this is the site of gluconeogenesis. Obviously, I'll be covering that in the biochemistry section. Next one is that it processes nitrogenous wastes. So that's basically processing urea. Detox of various wastes. I think just know that if anything is about detoxification in the body, it's smooth endoplasmic reticulum and the liver. Now, I obviously want to point out with that the liver has a lot of smooth endoplasmic reticulum in it. Like the cells of the liver, lots of smooth endoplasmic reticulum. It also stores iron and vitamin A. Another important role, I think this is if detox is important role number 1, I think this would be important role number 2. It would be the synthesis of bile. And it also beta oxidizes fatty acids to ketones. And the last one would be intraconversions of carbs to fatty acids to amino acids. So yeah, the liver is a very important organ in the body. I think the 2 main important ones would be that it's a detox center. It also synthesizes bile. And yeah, I'll add gluconeogenesis as well. That's very important for biochemistry. So yeah, those are the 3 important parts, or 7 important parts, but 3 being the most important of the liver. I think that covers everything I want to talk about with homeostasis. Like I said, this is a very detailed chapter. If you're confused about it, I think you should just read the chapter yourself and figure everything else out. Before I leave, I'm just gonna clean up the notes a little bit, so if you guys ever want to screenshot this, it would make more sense. All right, there we go, guys. And with that, oh boy, the endocrine system. let's start here. So I like this one because it's a lot more chill than homeostasis. So we can like sit back and relax for a little bit. No, I'm kidding. We're gonna go into detail. Let's start off with the difference between peptide hormones and steroid hormones. So peptide hormones, they act on the body via secondary receptors. So let's think G protein receptors, coupled receptors. And they are, since they act via secondary messengers, they do not enter the body, or they do not enter the cell directly. They have to be, they have to work with receptors on the surface. Steroids, on the other hand, they go directly into the body, or sorry, directly into the cell. They go directly through the phospholipid bilayer, and then they bind to the DNA directly into cell. And then they bind to DNA via receptors. Now, another thing I want to talk about with peptides and steroid hormones will be direct versus tropic hormones. Tropic hormones, I'll write it here somewhere. Tropic, they stimulate other glands. They go towards other glands and they increase hormone output through that way. Direct hormones, however, they just directly stimulate the organs themselves. So I'll just write straight to organ. Now let's talk about the various different hormones that are present in the body, in the endocrine system. This is every single thing that you need to know, I believe. Let's talk about the anterior pituitary, which will be your flag top. So who can tell me what flag top means? What is the acronym for that? Some of you guys might be using flat peg. I've always used flag top, but like whichever one works for you, use that one. Right, it's the acronym showing you like your where is it? Follicle stimulating hormone, luteinizing hormone, acetylcholine, blah blah blah. You get what it is. So no flag top. Those are the most important hormones present in your body. Also know what's going on in the posterior pituitary, which is just ADH and oxytocin. Your thyroid hormones, they're pretty easy. It's just thyroid hormone, which would be TH3 to TH4. Calcitonin. I'll go more into like calcitonin in the bones portion of this video. But yeah, just know that it does play a role in regulating calcium in your blood and your bones. Parathyroid hormone also plays a similar role. Thymosin. I think this is the only one that's a bit difficult, in my opinion, to recognize and memorize. So, thymosin. And I think the other one was ANF. Erythropoietin, that's not something that you really have to like focus on. You just know that like it seems pretty obvious that it's like associated with like red blood cell synthesis and bone marrow. But if it's something that you guys, I'll put it down anyways. Insulin, glucagon, somatostatin. Those are really important. Insulin and glucagon, I think a good way for it, you guys to remember is that glucagon is present when glucose is gone. So when glucose is decreased, glucagon is increased. It's secreted from the alpha cells, and insulin is secreted from the beta cells. So with insulin, obviously, it's just the opposite of glucagon. So when glucose is gone, glucagon is present. But when there's a lot of glucose present, insulin is present. It's decreasing the amount of glucose so that you can reset to your set point. Now, somatostatin, it just inhibits digestive processes. It also inhibits the secretion of insulin and glucagon. So somatostatin, I think that also might be a difficult one. Cortisol, aldosterone, these are your stress hormones. Aldosterone will be covered in the homeostasis section. So sex steroids, like it says right here, normally not important, but like estrogens for women or for females, Testosterone is for males. Progesterone is also for females. I'll cover that more later in this section. Now let's talk about feedback mechanisms. Now, what is a feedback mechanism, guys? Can you give me, like, explain what it is for me, or to me? Sure, yeah. Like a negative feedback loop would be like, so there will be something that will like inhibit the formation of a product. Yeah, sure, that works. I'll start with negative feedback loops. So that's basically what you said. Like a product is created that will inhibit the creation of more of that product. So an easy way for you to think about like a negative feedback loop would be, let me think of something. The thermostat is like the obvious like answer. I'm trying to think of like, I think the thermostat's a good analogy. So when your thermostat increases the temperature, or like when there's an increase in temperature in your house, your thermostat will be activated to create like air conditioning. So it would start decreasing it and trying to reach back to the set point. Note that there is no change in the set point, you're just trying to reach it again. Now with positive feedbacks, it's a bit different. Can anyone explain to me what a positive feedback is? Yes, it's like you increase the thermostat. It just keeps going up. Like it gets hot. It gets hot in the house. Your thermostat will make it hotter and hotter and hotter. So I'll be getting into that in just 1 second. But like the classic example of a positive feedback loop would be during childbirth when oxytocin is being released to like help the pregnant mother with her contractions. You need to be able to contract as much as possible so you can push the baby out. So during childbirth, it will be an increase in oxytocin, followed by even more oxytocin. Keeps on going until the baby's out. And another thing with positive feedbacks is that they're usually creating a new set point. So after whatever is happening, you reach a new set point. That's where your new base is. Now, let's finish this off with the menstrual cycle. There are 4 stages to it. The follicular stage, that's where FSH is being produced at a large amount. It's causing growth of the follicle. So right, like this part right here, let's say that's the follicular phase. Oh, great. Oh, sweet. It's labeled down here. Okay. That's the follicular phase. Ovulation is when a large amount of luteinizing hormone, this right here, large amount of luteinizing hormone is released, but causing the egg or the follicle to rupture, sending out an egg cell. And during the luteal phase, that's when like they start like leveling off, decreases in like luteinizing hormone, decreases in FSH, but there's a large amount of progesterone. Progesterone, what does progesterone do? I'll talk about progesterone and estrogen together. Estradiol is just a different form of estrogen. Estrogen establishes the corpus luteum. And if estrogen establishes, then progesterone protects. Progesterone protects the corpus luteum. It stabilizes it. It keeps it there. It also keeps the endometrial lining there as well. The corpus luteum plays a major role in forming the endometrial lining, which is basically where the egg will be located in order to grow. Now, menstruation occurs when the egg isn't fertilized. There's no sperm there to fertilize the egg, so we have to slough off the endometrial lining. We have to push everything out. So everything is just leaving the system. It's just everything will be returning back to normal as well. You can see that the hormones are like stabilizing here. Progesterone, estrogen, they're also stabilizing. And then it repeats. This is all a 28-day process, by the way. So this would be day like 1 through 14. Ovulation occurs on that 14th day, and this would be 15 through 28. A menstruation occurs and it goes through the same cycle all over again. So yeah, that was really fun, wasn't it, guys? I'm kidding. This is a really difficult pathway to memorize. I think you guys should memorize when luteinizing hormone spikes, when the FSH spikes, when is progesterone present, when is estrogen present. Oh wow, all that stuff is down there too. Yeah, this is a very important thing. I highly recommend studying this. This is probably the most important thing in the endocrine system to memorize, as it also goes with the reproductive system. So yeah, let's move on to my favorite chapter, the nervous system. It's so easy. This is so nice. So let's talk about anatomy first. I think all of this is pretty straightforward to you guys, right? Is there anything that you guys are confused about? Nothing. All right, cool. So yeah, obviously chemical electrical signals enter the cell through the dendrite, pass through the soma, reach the axon, are transported through the axon via nodes of Ranvier or myelin. They go through the axon terminal, and then they spread out towards whatever the axon terminals connect to. Now, let's talk about... Should I get covered impulse propagation first, or should I go straight forward towards the charts? I'll talk about the charts first. I'll draw it out myself. So, this is time. This is memory potential measured in millivolts, and I'll draw it. This. Can you guys see this? Something. Yeah, that's fine. All right, perfect. And I'll draw a little red line here. This is the resting potential. Resting membrane potential. So this is before, this area right here. I wish I had more colors. All right, that's fine. This is right here. This is before action potential is occurring. So note that action potentials are an all or nothing principle. So if you don't have enough of an action potential, or have enough strength in an action potential, you're not going to be able to trigger it. So if you don't have any action potential, it's not going to trigger it. But once you have enough, once you have a strong enough stimulus, it'll create an action potential, shooting up the membrane potential all the way up in a process called depolarization. Whoa. Yeah, there we go. Depolarization. Now, during depolarization, the sodium channels in the axon open up, letting all the sodium ions out. So it's rapidly depolarizing the membrane, going from negative 70 to about positive 30, I believe. The numbers aren't necessarily important, but the resting membrane potential is... Oh, I really screwed that up. No, this is not resting membrane potential, guys. I'm sorry. This is the threshold of action. This is resting membrane potential. I'm so sorry. That was a brain fart on my part. But yeah, it goes from negative 70 to positive 30. And then once you reach here, that's where the action potential occurs. And once there's no more stimulus, you're going to have to repolarize, causing you to drop all the way down. You're opening up potassium channels, letting potassium into the cell. That's what's letting it go all the way back down to -70. It'll dip a bit lower, actually. So like it'll be at -70. It'll dip even lower before restabilizing back to -70. That's a process called hyperpolarization. This plays a role in refractory periods. That's something you can read about in the Khan Academy documents for psychology. But just know that hyperpolarization and repolarization are different things, similar but different. Now, in hyperpolarization, I would just like to state that all the gates are closed. So in depolarization, sodium channel open, repolarization, sodium channels close, potassium channels open. And then hyperpolarization, they all close. And I guess at rest, I'll just say they're all closed as well. One thing I think I just have to cover is a sodium-potassium pump. It's not that complicated. Just know that with the sodium-potassium pump, it's located on the membrane. For every 3 potassium that is pumped out of the cell, every 3 potassium that's pumped out, every 3 sodium, 2 potassium are pumped in. All right, now let's talk about the synapse and impulse propagation. So at the synapse, this is a chemical process. That's where the neurotransmitters, when the active potential reaches the axon, it opens voltage-gated calcium channels, which allow vesicles to form and let neurotransmitters enter those vesicles and be pushed out into the synapse, where they will be pushed out. They will bind onto postsynaptic receptors on the dendrites, and they will be used to create whatever effect is needed. Usually, like, I think acetylcholine is something that you need to know. That'll be very important. This is how the dopamine reward systems work. This is obviously very tied closely towards your psychology section. So this will be covered in very good detail there. But yeah, that's basically it. Note that's a chemical process, while the propagation of the action potential through the axon itself is an electric process. This is done by saltatory conduction. Which allows the action potential to rapidly shoot from one axon, or one node of Ranvier, to another. And this is sped up mostly by the presence of myelin. This is basically just this is what makes, it's like a fatty acid sheath. This is what basically, when you see gray matter and white matter, this is what makes white matter white. So yeah, this allows rapid propagation of signals from axon to the terminal, and allows it to be transported very quickly throughout the brain. I think that's everything I need to cover with the nervous system. I think everything else would be like central, peripheral. I think you guys know that stuff. Peripheral goes into sympathetic, parasympathetic, all that stuff. I think you guys should be able to. You guys know that stuff, and if not, go through Khan Academy's 300-page doc. Oh boy, the musculoskeletal system. This is my least favorite chapter. Where do I even start? Let's go here. So this is the sarcomere. It's like the functional unit of the musculoskeletal system. Note that the various like areas of the sarcomere. So the I band, I'll write it aside. The I band, it only has thin filaments. So these thin filaments are actin. While the H zone, you guys see, oh, it's not drawn here. I'll draw the H zone. This right here, that's the H zone. H zone, it only has thick filaments. Those thick filaments are myosin. Anything else I want to cover? I guess like the A band. So when you use your muscles, they contract. Your sarcomere contracts. It goes inwards like this, right? You see right here, like this will be pushing itself inwards. So everything in this diagram is contracting with the exception of the A band. So I'd like to note that the A band stays constant during contraction. Yeah, I guess that's basically everything you need to know about the anatomy of the musculoskeletal system, at least with the sarcomere. Let's move on here to, let's move on here towards the contraction and relaxation of the muscle. So during contraction, depolarization leads to the active potential leading towards everything that's occurring. That's when the sarcoplasmic reticulum releases calcium. So, by the way, like sarcoplasmic reticulum is analogous to the endoplasmic reticulum in normal cells. It's just called sarcoplasmic reticulum in muscle cells. So yeah, it releases the calcium. The calcium will bind. The calcium will bind troponin to actin. So that troponin right there, it's going to bind to actin. And then that will cause a shift, which will expose the myosin head. And that exposure will cause the ATP to bind, and it will dissociate, and it will eventually cause it to move. The myosin and actin will move. Now, relaxation occurs when the calcium is pumped back into the sarcoplasmic reticulum. So then the ATP will dissociate, it'll go back to where it was before. Everything will return, and it'll wait until the muscle is being used again, and it'll go through the same process. I know that was a bit weird of an explanation. It's kind of bare bones. Like I said, this is like similar to homeostasis. If you don't know it, you genuinely have to just read the passage again. Good news is this isn't that deep. Like, it's not like something you'll be like deeply tested on, and they'll probably give you the concept in the passage if you need to. So memorize this. I think it's pretty straightforward. I'm going to be moving on to bone formation and bone remodeling. So I'll just say right here, osteoblasts build bone. That means calcium is being released into the bone. Osteoclasts cleave bone. Now, what does that mean? It's kind of like the reverse thing. This is blood calcium, by the way. So all the calcium that is present in bone is being released into the blood. It's like they're basically like reverse of each other. So reformation is when ions are absorbed into the blood for use in bone. While degradation is when or ions in the bone is being released out. So they're similar to blast in class. I'm trying to think of a better way I can like write this for you guys, because this will be tested on the exam, and this is very easy, so I want you guys to get this right. It's slipping my mind right now, guys. I think blast, build bone, class, cleave bone is pretty good. If it ever comes back into my mind, or if you guys have anything in chat, feel free to say anything. If not, let's just move forward from musculoskeletal to the circulatory system. Now, circulation. Actually, how long have we been doing this? I'll finish the circulatory system, and then we'll take a small break, and then we can come right back. All right. So circulatory system. Now that it seems straightforward, but every student I've worked with has always struggled a little bit with the pathway through the heart. So I'm just gonna say it really quickly for you guys. It's inferior vena cava, right atrium, or I'll write it down. Inferior vena cava, right atrium, right ventricle, pulmonary arteries, lungs. And the lungs go to the pulmonary veins. Then they go to the left atrium, left ventricle, this pathway is a bit weird, I know, I'm sorry guys. Aorta, body. So after it goes to the aorta, it goes wherever the body needs oxygen. And then whenever all that deoxygenated blood is being used up, it goes all the way back to the inferior vena cava. You guys should write this in a better way than I did. But like I don't know why I didn't give myself enough room, but that's basically the gist of it. Here's this looks a lot more complicated than it does, but this basically covers everything that you need to know about circulation. One thing I'd like to talk about is fetal circulation. It's a bit different. These are mostly just vocab words, so I think you guys should look this up. Not fetal circulation. Foramen ovale, ductus arteriosus, ductus venosus. Those are basically just vocab terms you guys need to know. Know that the foramen ovale connects the right and left atrium. This isn't the fetus, by the way. This is not present in adults or even children. The ductus arteriosus and venosus, they kind of work similarly. The arteriosus connects the pulmonary artery to the aorta, and the venosus connects the umbilical vein to the inferior vena cava. I don't have enough room to write all that stuff down, but these are basically just vocab terms. Look it up. Make flashcards out of them. Yeah. So moving on, let's talk about blood really quickly. You guys know your red blood cells, you know your white blood cells, you know your plasma, platelets, you know all that. I'd like to talk specifically about the Bohr effect. So this is what it is right here. Now, the Bohr effect is when there is a decrease in pH, which leads to an increase in the CO2 pressure. Now, that's basically what this says right here. I'd like to say that in addition to just a decrease in pH leading to a rightward increase or rightward shift, rightward shifts also are caused by an increase in temperature. decrease in pH, increase in PCO2. I think the best way to talk about this is by talking about this equilibrium equation right here. CO2 is H2O forms bicarbonate, leads to H plus. Oh, I ran out of room. That's embarrassing. Let's move it a bit further back. H2O plus CO2 yields carbonate. H plus HCO3 minus. So when there's a decrease in pH, that means there's a decrease in the hydrogen ion concentration, right? So if you have a decrease in the hydrogen ion concentration, note going through Chatelier's principle, I'll talk about that in chemistry lecture, of course. You would be increasing the amount of your reactants. You don't have to like worry about this. It's just an intermediate. You increase the amount of reactants present. Water is a liquid. You don't really have to worry about that. So the only thing that you have to be increasing will be PCO2. So there's more of that when there's a decrease in that. Let's think about it in the way of temperature. If there's more temperature, why would we cause a rightward shift? Right, temperature is always seen as a product. So temperature, you would see it as like a product here, causing a shift towards more PCO2, leading to a decrease in pH, leading to a rightward shift. So yeah, I think that pretty much covers everything I want to talk to about the Bohr effect. Make sure you know why everything is moving towards the right or why things will move towards the left. So like an increase in pH, decreased temperature, less binding of oxygen, of hemoglobin. I'll talk about the hemoglobin stuff in the biochemistry lecture because it's a really good example of cooperative binding. So let's talk about blood types. I'm going to see what I can like say about blood types because it seems pretty straightforward, right? What exactly can I say? It's essentially a Punnett square situation. Just know that AB is the universal recipient, O is the universal donor, RH positive, and RH negatives. RH negatives, they don't have the RH antigen, but the RH do. Just know this chart. It's an easy thing that'll show up on the exam. I highly just know the chart. You guys should be fine. With that said, what do I have for respiration? Yeah, let's go through respiration and then we take a break. Sorry, guys. Respiration, then a break. So respiration, there's not that much going on. Or there is, but like you don't have to know it for the exam. So we know the pathway, right? It's pretty, go through the nasal cavity, nostrils, or the oral cavity. It goes to the pharynx, larynx, epiglottis is more associated with the digestive pathway, but like you know what it does. It goes to the trachea, through the bronchioles, superior, inferior lobes, and all that. Eventually, it will reach the alveoli. This is the site of gas exchange. And then we eventually just breathe in. Oxygen is entering the body. Carbon dioxide is being pushed outwards. One thing I'd like to point out is that the contraction of the diaphragm when you inhale is negative pressure breathing. So that's something I always struggle with. I'm not entirely sure why, but I just never got it. I want to make sure you guys get it. So contraction of the diaphragm. Negative pressure breathing, that's associated with inhalation. Now, obviously, relaxation of the diaphragm, that leads to, that's a bit, and that's obviously exhalation. You push everything out. One thing I like to note is that fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin, and that the gas exchange occurs there in the placenta. Now, if there's any one problem that I'll talk to you about respiratory, I will have something to do with this right here, tidal volume and all that. So I think this is pretty straightforward once you get to know like the various terms. So tidal volume is like you're basically like your breathing pattern, like you're in and out. So when you breathe in, this would be increasing your volume, decreasing your volume when you exhale. So inspiratory reserve volume is your maximum inhalation. So when you puff yourself up like that, when you breathe in as much as possible, that's how much air is in your lungs, have the maximum amount of air in your lungs. Now, expiratory reserve volume, it's sort of similar in that when you exhale, your lungs release as much air as possible. But it's not like you can go all the way down to zero, because if you do, your lungs will collapse and you won't be able to breathe in again. So that's why the minimum is like, say you're saying 15. It could be different for like a child, like an elderly person, but like for this one, it says 15. Functional residual capacity. This is the functional amount of like air that you have after you exhale. So you have to take in more to reach tidal volume. Vital capacity is how much you're holding in, minus the obvious like air that you need in your lungs in order to prevent collapsing. Residual volume, pretty self-explanatory. Inspiratory capacity is similar to residual capacity, and then total lung capacity is the maximum amount of air that you can put in your lungs, like just in general. So yeah, memorize this chart. If they ask you any questions like, what would this be, or like what is causing someone's expiratory reserve volume to decrease, just memorize this and you should be able to figure it out. Oh, we're taking a break on a good part because the immune system's pretty deep. I'll try and get. All right, there we go. Let's say 5 minutes and I'll be back with you guys shortly and we can figure this out. Alright guys, you guys ready to get back into this? Yeah, sorry, I was checking to see if I was actually recording. If I saw that I was recording for like an hour and I felt like nothing was actually like being, or if I was just like talking into my screen for an hour, I'd be like really frustrated. Yeah, I know, chat was there. Alright, let's get into the immune system then, guys. So, immune system, this is a very dense chapter. This is also like one of those chapters where if you don't know it, you just have to read it. So, immune system, musculoskeletal system, homeostasis. Those three, you just have to just read it, get to know it. Because the immune system really is important. It's the most important, I wouldn't, the cell obviously is more important chapter, but I think the immune system is the most like important like body system chapter out of all. So let's just get into it. Let's talk about like what an antibody is. You guys are seeing right here. There is a constant region and then a variable region. The heavy chain to light chain. The constant regions are the ones that stay the same. Variable regions change depending on which antigen they want to bind. So this is what's going to be changing in order for you to bind a specific antigen. I'd like to note that every antibody and antigen pair is very specific. Yeah, okay, so my mic is on. Yeah, so everything is very specific. It's similar to like how like enzymes work. It's very like one-to-one. So let's move to, do I have anything written here? I don't think so, right? It's just the stuff from the previous chapters. Specific defense. Let's talk about beta cells. This is also known as Alright, yeah, no, we can start with beta cells. So beta cells, they can be split into memory cells or plasma cells. Now, memory cells, they're pretty like straightforward. They're the ones that memorize the antigen. They speed up the secondary response that occurs in the future. Plasma cells, on the other hand, they are the ones that make and release antibodies. They're the ones that induce antigen phagocytosis. They're specifically the antibodies that you should memorize are IgG, A, M, D, and E. Their functions aren't necessarily important. Just memorize that like these are the most important ones. What else do I want to talk about with beta cells? Beta cells, like their specific thing is called humoral immunity. Yeah, all right, there we go. So yeah, their specific thing is called humoral immunity. That's a specific defense. Another specific defense comes with your T cells. Cytotoxic CD8 cells, I believe. Oh man, that's gonna eat me up. I'm gonna double check for you guys real quick. Yeah, I think I'm right. These are CD8 cells that are cytotoxic. CD8 plus cytotoxic C cells. Yes, sorry about that, guys. That was just a weird brain fart on my part. These destroy cells directly. Helper T cells, these are CD4. And these they activate the beta or the B and T cells as well as macrophages. And the last T cell that you want to know would be suppressor T cells. Now these they're the regulatory parts of the T cell, or the T cell family. They regulate B and T cells, and they decrease antigen activities. I'd like to point out that these are cell mediated. Instead of humoral. Now, these are both specific defenses, non-specific defenses. These are like typical like skin, sweat, passages that are lined with like cilia, your inflammatory response, stuff like that. It's something that I think I need to write out and like have like a conversation about. Let's talk about the lymphatic system. There's nothing much that I want to talk about the lymphatic system really, but like it always shows up. It's something that you need to know. Your vessels carry lymph, and yes, I think like lymph vessels. I'm trying to like write this out as a pathway for you guys. So lymph vessels, they go to your lacteals, and your lacteals are like are portions of your body where they collect fat, and they're located in the small intestines, and they are absorbed by chylomicrons. So your lacteals collect or whatever they get from lymph vessels, and they transport it to the lymph nodes. Or I wouldn't say that they are transported to the lymph nodes. Lacteals are like the where they end. The lymph vessels transfer it to the lymph nodes. Then they eventually all end up in lacteals, where they are absorbed in the small intestines, and then they're excreted out as waste. There's a lot of like anatomical stuff with the immune system. I think is important. Actually, no, never mind. I don't think so. I'm thinking about the digestive system, which is the next topic, which we can just go into right now. Yeah. Digestive system. Where do we start? Let's start with anatomy. So obviously, food enters through the mouth, gets processed, they get masticated by the teeth and the tongue, enter the pharynx, esophagus, stomach. They go through the duodenum, jejunum, and ileum sections of the small intestines. I think an easy way to memorize that is Dow Jones International, which is duodenum, jejunum, ileum. And then they enter the cecum of the colon. They ascend of the ascending colon, the transverse descending. This is the large intestines, also all known as the colon. They enter the sigmoid colon for storage, I guess. Not I guess. Sigmoid colon is the last portion of the colon where it's secreted out to the rectum, where it's then stored, and then it's released through the anus. So yeah, where do we go from here? Let's talk about. I have all this stuff right here. These are all the various different enzymes that help with digestion. They all have a very specific role. I think the most important one to know would be salivary amylase. That's the one that's present in the mouth and in your saliva. They highlight some here, but I think like some that you absolutely have to know will be pepsin and its zymogen pepsinogen, aminopeptidase, bile I think is very important, lipase is important. I'll get into lipid digestion in 1 second. Trypsin's also important, and I guess trypsin as well. Yeah, I know I start half of it, but those are the ones that you guys absolutely need to know, memorize. Sucrase, lactase, they seem pretty obvious. But if you guys struggle with that, look into that as well. Yeah, sorry, my pen just started glitching for some reason. Now let's move on to lipid digestion. So, how do I want to say this? When chyme is present, your duodenum secretes a hormone called CCK. Cholecystokinin is secreted into the blood, and this increases the secretion of pancreas enzymes. And it also increases bile. So, like I said before, bile is made in the liver. It emulsifies. The purpose of bile is that it emulsifies fat. In the small intestines. Do you guys mind if I just write S intestines, or do you want me to write out the entire thing? S intestines is fine. All right, cool. So in the small intestines, I think the one thing you want to know is that it's not an enzyme. Yeah, I write so fast. So yeah, cholecystokinin, it promotes the increase of bile, emulsifies fat in the small intestines. I think we should also talk about lipase. Lipase is also made in the pancreas, and it hydrolyzes lipids. In the small intestines. Protein digestion and carbohydrate digestion, I think they are covered pretty accurately here. If you guys have any questions, I'd be feel to answer. I'm not really sure what I can cover right now. Yes, I think definitely like reading the chapter would do you like wonders with digestion. Actually, no, never mind. Digestion isn't covered that much in the. Well, if it's something you're struggling on, sure, you can reread the chapter or whatever. But like, I think like this shouldn't be like number one on your list. You know, I still think that like the immune system, the musculoskeletal system, and the cell are much more important. So this would be like, I guess, number 4. That being said, I think we should just move on. All right, guys, we're almost done. We're reaching the easier parts, genetics. Genetics. So let's start off with, you guys are familiar with genetics, right? Like Mendel's laws and all that. If not, I have it written up here. Law of segregation means that each gamete has one copy of each gene. Law of independent assortment, I think it's just easier to explain with Punnett squares. So let's just do some Punnett squares, right? Let's do one quick one for the sake of doing one. So this is a heterozygote mixed with, I'll do homozygous recessive. So you get A here, A here, and then here it's just A. Meaning that you'll get 2 heterozygous children. So, or there's a 50% chance you'll get a heterozygous child. So they will still have the dominant trait, or they will still show the dominant trait, or they will have 50% chance of getting homozygous recessive, showing the negative trait. So I think one that always confuses people is whenever like you get something that's like A, B, B, something like that. This is very common on the MCAT, by the way, or like something like that. So you'll get like something like R, X, or like X and Y specifically for like sex chromosomes. So I'll just do like Z, something like that, right? So these kinds of Punnett squares, you don't bother trying to draw it out. I think it takes a lot of time and a lot of thinking for something as simple as a Punnett square. Just know that the phenotypic ratio would be like 9, 3, 3, 1. So this would be like completely dominant, this would be completely recessive, and these will be like heterozygous, with this one being dominant, one being recessive, and like vice versa. The statistics of I think you guys know that one. It's like they're all independent of each other. That's why like independent segregation. You guys should know this stuff. Let's move on to genetic mapping. I specifically marked out this one because I want you guys to figure this out. So genetic mapping is when the genes are separated and you're trying to figure out the recombination frequency. The more spread they are, the less likely they are to be or the more spread apart, the more likely they are to be unlinked. Genetic mapping, this occurs during prophase 1, by the way. So I just like to point that out. I think another thing I like to point out is that the percentage that you'll see here is like 17%, 8%. 1% is equivalent to 1 centimorgan. So it's the equivalent, and that's basically the fundamental unit. Now that being said, if this is 8% and this is 17%, what do you guys think this is? Go on. It's not a trick question. You guys, this is pretty straightforward. Right. So it's 17 minus 8. So that equals 9. This is 9%. And that's how like a question like this would be solved on the MCAT. It's pretty straightforward. I wish they would give you something this easy though, but like the gist of it is like once you get down to like figuring out what they're asking, it's as simple as this. Patterns of inheritance. I have that written right here, right? Yes, I do. Let's talk about that just for 1 second because this will always show up. The good news is they don't talk to you about like what I forget, it's slipping my mind, but like those family trees, like stuff like this, they won't talk to you about that. So let's talk about patterns of inheritance. So autosomal recessive inheritance. These are the ones that will skip a generation. Autosomal dominant, though, these are the ones, these are like your standard ones, guys. These show up every generation. And then the last one, I think, let's talk about like sex-linked. Sex-linked is like it shows up on the X allele only. So there is no, it's not like male-to-male transmission. But instead, it's like, I think a Punnett score would be perfect to explain this. You're right. Let's move a bit here. So let's say we have a woman that is a carrier for a sex-linked disease. and let's say we have a man that doesn't express it at all. So if they have a daughter, there's a 50% chance that she doesn't have it at all, or there's a 50% chance that she carries it. Because this right here, this is a recessive trait. But with the male, the sons that they could have, there's a 50% chance that he won't have it at all, or there's a 50% chance that he will have the disease. So because of this, with sex-linked gene or sex-linked diseases or any traits, they always are present with the male. So like, if I change this to, even if we do it like this, we 100%, or no, sorry, I moved too fast. It's a 100% chance that a daughter will be a carrier, but still, like, the son will be the one that's expressing it. Now, I think a good example of that, or the first example that comes to my mind of a sex-linked trait would be colorblindness. That's why you see like most colorblind patients are male. Now, moving forward, let's talk about molecular genetics. Let's look at the anatomy or like the structure of a nucleic acid. This is a nucleotide, which is the sugar, the nitrogenous base, the phosphate group. DNA sugar would be deoxyribose. This right here is RNA because of the ribose present. DNA would have deoxyribose. I want to talk about purines versus pyrimidines. So purines, the ones that we are considered purines would be adenine and guanine. Pyrimidines are cytosine, thymine, uracil. I think a good way to memorize this is that there are only 2 purines, but 3 pyrimidines. Like 3 isn't a pyramid. Like, yeah. So I think that's a good way to memorize that. Another thing I want to say is that purines always bond to pyrimidines. So that's why like A and T go together, G and C go together. I should have written this out in a more like logistic way. T plus U plus C. So it's like that. One more thing I'd like to say about that is that A double bonds to T while G triple bonds to C. Let's talk about... I put mutations here, but I think I sort of like took too much space. I'll move it. We'll talk about it later. We'll talk about viruses first. Actually, no, I think it would make more sense to talk about mutations first. So let's talk about mutations like right here. So mutations are when like a nucleotide is changed in some way or substituted or deleted. So we can talk about it in different ways, such as point mutations. Now these are your substitutions, like I said. Your silent mutations. Silent mutations, nonsense mutations. Now substitutions are also known as missense, because like that's when you switch one out. Silent is when you sub out one nucleotide for another, but it doesn't change the nucleotide or the amino acid. There's no change present. Substitution with a missense usually causes like a change to form in the amino acid, so a change in the amino acid. And nonsense, that's when it causes a premature stop codon. So that would be your UAG, UGA, And then another kind of a mutation is called a frame shift. Now these are a bit bigger, I believe, in that they insert or delete from the reading frame. So these always cause, like, the protein doesn't form at all. Or it's non-functional. So yeah, I think everyone memorizes the nonsense one pretty easily just because it's a very dramatic one. But like silence, just remember that there's a substitution with the amino or with the nucleotides that doesn't cause the amino acid to change. Missense substitution, it will change the amino acid, but like it won't be that big of a deal. So yeah, I think that's it for mutations. You don't have to worry about like memorizing like all that, like UAG and UGA as your stop codons. Like that's something you have to memorize, but like for everything else, they'll give you the chart or they'll tell you what it is. You should be fine. And now let's talk about viruses. So viruses they're not alive. They're acellular, and they can have either double stranded or single stranded DNA, or double stranded or single stranded RNA. I think you guys know the structure of a virus, like the capsid, the bacteriophage, like anatomy and all that. It shouldn't be that difficult. I'll just cover like lytic and lysogenic cycles. So this stuff, it's pretty straightforward. So with the lytic cycle, the bacteriophage infects the cell, inserts its own DNA inside the cell. It doesn't mingle with the host DNA. It doesn't add itself in there. Instead, it just circularizes and stays separate and begins replicating on its own. So it creates more bacteriophages, and it just keeps on increasing over and over again until it lyses the cell when there's too many. It lyses the cell and it leaves. And it begins infecting more cells. So I'd like to give you guys an example of a lytic cycle, a lytic cycle virus. Can anyone think of one though? Does anyone have any ideas? Okay, so none of you guys have an idea. The idea I want to give you guys with the lytic cycle is that it's a very, very fast process. Bacteria comes in, rapidly replicates, kills this host cell, goes to the other ones, keeps on going until it just burns out. It's a fast one that goes out of your system within 2 weeks. You know anything else that goes out of your cell into your body in 2 weeks? Yeah, COVID-19. That is an example of a lytic virus. Now let's move on to lysogenic. The lysogenic is a slower paced one. The phage infects the cell just like it does with the lytic cycle, but it inserts itself into the genome. Now, as the cell, it lives its normal life, it goes through mitosis, it divides, but the genome is present in the daughter cells. It keeps on replicating like that over and over again until it reaches some sort of stressful situation, where then the phage DNA is excised. It removes itself from the host DNA, and then it goes right back into the lytic cell and creates more. So this is a long-term one. This one happens over time. You might not even know you have this. Does anyone have any examples of this? Would it be STDs? Yes. My example was actually herpes. But yes. I know some of you guys are saying HIV, and I think that is a very good example of a lysogenic cycle, or lysogenic virus, but I would rather think of HIV in the terms of retroviruses. Retroviruses, they use reverse transcriptase. They change from the RNA to a DNA. So that's something I like to cover in biochemistry. But yeah, HIV, think of it as a retrovirus. Lysogenic cycle viruses, I think of it just as STDs in general, like herpes. Lytic cycle, COVID-19. It would be really funny if this actually showed up on an exam. I don't think it would, but if it did, it would be really funny. This one right here is just the genetics of prokaryotic cells. These are prokaryotes. I think this is pretty straightforward, you guys. I think you guys know what this is. Transformation, that's when a bacterium inject or acquires genetic material from its environment and then it adds it to its own genome. Transduction is when a bacteriophage like introduces it. It's kind of like the lysogenic cycle, but obviously it won't lead to something like this. It just increases its like genomic code. It just makes it it like adds to the bacteria. And I think conjugation, this one is the most important one because it's obviously the most complicated one. Conjugation is when two bacteria, one which has a sex pilus attaches to the one without the sex pilus, introduces its DNA into the other bacteria, and it adds it to its own genomic code. Any questions? All right, cool. Let's wrap things up, guys. This is the last section. Yeah, last section. Cool. Evolution. I'd like to start off with natural selection. Oops. Let's start off with natural selection. We're all familiar with Darwin's theories on natural selection. This one right here, I'd like to call this one, or this one is called stabilizing selection, where one trait isn't necessarily preferred, but instead a mediating trait is preferred. So like if we use like that, like Darwin's finches, you don't want like a beak that's too big that can't enter the wood to pick out the seed, but you don't want a beak that's too small so that you can't that it's not long enough to enter the wood. You want like a medium-sized beak. Similar or like in the opposite concept, it's disrupting selection, where like you don't want the mediating one. You want either a really small beak or a really large beak, depending on where like you're living. And then this one's directional, where like one is obviously preferred more than the other, instead of disrupting where two are preferred or like the extremes are preferred. So if you ever see like a chart, I highly doubt you will, but like if you ever do, like these are the ones that you guys should be thinking about. Now let's move on to divergent speciation, parallel, convergent. Now divergent speciation is when two separate traits are developed from two similar animals. So I think one that I think you guys should think about would be divergent traits. Let's think about like seals and dogs. How about that? So seals and dogs, they're very similar mammals when you think about it, but the difference that comes with them occurs through divergent evolution, where seals, their flippers, their limbs are developed to swim. So they obviously prefer to swim. They're designed to swim, while dogs' limbs are for the land. So they run, they hunt on the land. So they have very similar like features anatomically, but because it's divergent speciation, they instead have to use their limbs separately. Now, parallel speciation is when two species over time independently develop the same trait. So I'm trying to think of a good example of that. I think it's mostly just like two different birds would develop the same trait. Like two different species of Darwin's finches, if they're present on the same island, they would want to get like a certain size beak in order for them to reach the seeds that are present in the wood. And lastly, convergent. It's kind of like it's the opposite of divergent, where like one trait from two separate species is developed by both. So one would be like fish and dolphins. All right, just birds for a parallel. Fish and dolphins. So fish and dolphins, they're not they are one's obviously like one's a fish, one is a mammal. So they have different structures, or they have similar structures actually, because they developed in the same environment, but they function differently, as one is a mammal, one is a fish. They're from different families and all that. So moving on to the last thing I want to talk about is Hardy-Weinberg equilibrium. Now, there are 5 traits there are 5 conditions you need to meet for Hardy-Weinberg equilibrium, and that would be no mutations, large population, random mating, no migration, and reproductive success. They'll usually give this to you on the exam, but if they don't, like, you need to realize that like all of these must be happening. This could be like a question, be like, which one of these is not necessary for Hardy-Weinberg equilibrium, and they'll give you like 3 answers from this and like 1 answer that isn't from one of these 5. So now moving on to the actual equations of Hardy-Weinberg equilibrium, we have P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. So I want to stress that the P and P squared are not the same. They're completely different variables. P means it's the frequency of the dominant allele. So how much of say A is present? Q is the frequency of the recessive allele. So P squared is the frequency of dominant homozygotes. So P, I think you better write it here. So this would just be like A, this would be A, dominant homozygotes. Q squared, frequency of recessive homozygotes. So this would be your AA. And now, obviously, 2PQ would be the frequency of heterozygotes. So A. So yeah, I've always gotten really tripped up over these just because I was thinking P is the same as P squared. So just know that P and Q are associated with the alleles. So they're only one letter. While P squared, Q squared, 2PQ, they're associated with the final phenotype. So homozygotes, heterozygotes, dominant, recessive, all that. With that being said, I believe we're done here. We covered basically everything important for you guys. Is there any are there any questions you guys have about this or any future lectures that I'm going to have about this? I'm going to get these out for you guys by April 9th. or April 7th. I want you guys to have some like rest before the exam. Yeah, if you have any questions, make sure you check out the videos I put on my website. Those are the ones that are going to be like super in depth. I have practice problems for that as well. So yeah, don't worry if there's like, if you think I went too fast with this. It's just supposed to be like a big like cumulative review covering like the most important stuff. This is like roughly 90 to 95% of the stuff you'll see on the exam. So yeah, no, if you have any issues, anything that you're just like struggling with, check out my videos. I do practice problems too. I do in-depth explanations. If you have the Kaplan books, they honestly work pretty well too. I think Kaplan's probably your best bet. I mean, honestly, I shouldn't be saying that like 1 week out. You guys are obviously do have books. Yeah, if that's it, I'm glad you guys stuck around. I'm happy to help. I'm ending stream right now, so if you guys have any questions, feel free to leave comments, or I'll leave my information in the description. We can talk about it, and I'll see you guys tomorrow with my lecture on. This was pretty dense. I think I'll go with the chemistries tomorrow. I'll do general chemistry and organic chemistry tomorrow. All right, sounds good, guys. I'll see you all tomorrow then.